very fast. No matter what we do, I feel like November is gone and the boys are going to. So we're basically already at the end of the semester. You just don't realize it yet. All right, so it is November 1st. You guys had a good Halloween last night. The ghouls and the goblins stayed at bay long enough for you to get what you needed to done and everything else. All right. So today we need to continue our discussions from class on. You remember on Wednesday we've been talking about kind of this blood-air interface and this blood-tissue interface where we have the diffusion of either oxygen and carbon dioxide happening, right? So in the lungs, right? In the lungs, we had a situation where the concentration gradient, this is our alveolus right here. And then this is the blood over here. This is the blood right here. Right? This is our blood here. We had a situation where we had oxygen diffusing from the alveolus into the blood because of its concentration gradient. And CO2 was doing the opposite, right? CO2 was diffusing from the blood into the alveolus for us to exhale as essentially metabolic waste. Okay. At the level of the tissues, we saw the opposite occurring. Okay. At the level of the tissues, we saw essentially the opposite occurring. So this is your tissue. We're using that term pretty loosely. Remember that we could have it could be muscle, it could be digestive tissue, it could be just about anything, and then we have the blood. But because the tissue has been using oxygen, the concentration gradient favors the movement of oxygen from the blood to the tissue, and that metabolic waste, CO2, that's been building up in the tissue is now the concentration gradient favors it going into the blood. So it's essentially the exact opposite sort of a situation. But we said on Wednesday that it's really just concentration gradients that are driving this, and this is just simply the diffusion of gases from one space to another, right? So, in terms of the mechanism by which these gases go, it really doesn't get more simple than this, right? Diffusion is one of the very first things that we learned about when we started studying biology. And this is essentially what it comes down to. It's just diffusion and concentration gradients, okay? But the question that we want to address today is, in the blood, how are these gases carried, right? Because in the blood, we have to carry both oxygen and CO2. And how are those being transported in the blood itself? Okay. So probably, let's just go ahead here and just write on the board, right? So we'll say here, how are oxygen and CO2 transported in the blood? You guys kind of already know the answer to this because we alluded to it when we talked about the circulatory system, but we want to address it more directly today. But how are oxygen and CO2 transported in the blood? Okay. Well, the most obvious solution for this would be that we would just simply dissolve those gases in our blood plasma, right? So dissolve them in the blood plasma, right? So we have options. Okay? We have options. And one of them is dissolve in blood plasma. Okay. Let me know factors that affect gases dissolving in solution. You had chemistry, you might have talked about this. temperature's one for sure, right? So when we look at solubility of a gas, let's come over here really quick and talk about solubility of gases in solution, right? Solubility of gases in solution. If we take a look at this, one of the things that matters big time is temperature, right? So temperature does affect how much gas can dissolve in a solution. Remember, if higher or lower temperatures increase solubility of a gas. Sure. Lower temperatures, right? And when a liquid is cool, you can actually dissolve more gas in it than when the temperature is higher. Okay? So cooler temperatures actually increase the solubility of a gas. Okay? This kind of a non-human biology analogy to help you remember that, right, is that one of the consequences of global climate change, of course, is that large bodies of water are increasing their temperature, right? They're starting to warm up. That means that less oxygen is dissolving in those bodies of water, and that's affecting fish populations, right? Because they extract their oxygen from the water. If we don't have as much oxygen dissolved in that water, they're not going to get enough oxygen to survive. So cold rivers and lakes and streams and things like that are really important to maintain ecosystem health because we're dissolving more oxygen in that water for those aquatic organisms. Okay? So that's kind of a non-human analogy to maybe help you remember which direction is which. And this is a little counterintuitive. I feel like in biology we need to warm everything up, but we can't do that, right? So temperature is one of them. Another one is, of course, is pressure gradient, right? So pressure is another one. Okay. So now, if I increase or decrease pressure, am I going to be able to dissolve more gas? To increase temperature, right? Or increase pressure, right? So under high pressure, I can actually dissolve more gas in a solution. And you guys have definitely seen this one firsthand, right? You probably have all at some point had a soda, right? I'm thinking about like a bottle of 7-Up or something like that, right? When you get that bottle of soda fresh, and 7-Up's a good one because it's usually under really high pressure. But when you feel the wall of that bottle of soda, it's like there's a lot of pressure there, right? And then what do you do when you open that bottle, right? You open that bottle and you release the pressure. And what happens to all of that gas that was dissolved in solution? All of a sudden you get bubbles in your soda, right? Because all of that gas is coming out of solution, right? So all you have to do to kind of visualize this one is just go buy a 7-Up and we'll be able to see it firsthand, right? So temperature and pressure are both important factors, and they can definitely increase the solubility of the gas in solution. And then the third and final thing that I can think of anyway is that every gas, whether it's oxygen or carbon dioxide or nitrogen or anything, kind of has an inherent quality of itself that's called the solubility of the gas, right? Some gases are simply more soluble in solution than others, and that's just an inherent quality of those gases, right? So I'm just going to put solubility of the gas. Solubility of the gas. And again, some are just simply more soluble than others. Okay. So let's go ahead and go through some of these. So how could we alter these to maybe affect how much gas we can carry in our blood? The reality is we can't change temperature a whole lot, right? You and I are what we call homeotherms. We maintain a relatively constant body temperature within a couple of degrees. So there's no real way that we could go into a hypothermic state to dissolve more oxygen in our blood, at least not at the cost of other things, right? Like a lot of other things would suffer if we tried to do that. But I suppose we could, go into a hypothermic state and get more oxygen into our blood. The problem is that other organs would shut down. So temperature doesn't really affect us that much because we do maintain a constant body temperature. Pressure is another one that doesn't change a whole lot for us. You have to change pressure pretty significantly to really affect the solubility of a gas. Increasing pressure is kind of the big one here that we would need to do. Being at sea level, or very close to sea level, we can't increase our pressure very much, right? The only place we can go from here is up to the mountains, and we know that pressure decreases when we do that. So it's not really going to affect us a whole lot. The only place where this could affect you, does anybody scuba dive? Anybody ever scuba dive? No? You should scuba dive sometime. It's kind of cool. I've heard. I'm afraid of the ocean, so I've heard it's cool, but I don't know for sure. I've snorkeled and I enjoyed that. But scuba diving, one of the things that you have to pay very close attention to is when you go down in the water column, you have to be really careful with how fast you come up, right? So one of the things that water does that air doesn't do is it exerts a lot more pressure on us. It's just simply heavier than air. So you don't have to go very deep underwater to have a lot of pressure exerted on you, okay? So when you scuba dive, you actually can increase the pressure on your body significantly, okay? And when you do that, what happens is that you're breathing this gas, and all of this nitrogen gas is going into solution in your bloodstream, okay? If you come up too quick, right, like if you see, a shark, right, and you straight up towards the top of the water, okay? If that happens, what's going to happen?
Decompression sickness.
Well, it is called decompression sickness, but physically what's happening?
Not enough nitrogen.
Well, all that nitrogen that you've been breathing that's gone into solution in your bloodstream is now going to come out of solution because you release the pressure almost instantly. So you know those bubbles that appear in a bottle of 7-Up when you open it? That's what happens in your bloodstream. All those nitrogen bubbles pop out of solution in your bloodstream, okay? It's called decompression sickness or the bends. And what can happen is that those bubbles can actually lodge in your joints and they can wear away at articular cartilage and do all sorts of really damaging things, right? So the way that they treat you for that is that they put you back into a pressure chamber, they put that gas back into solution and bring you out very, very slowly to avoid that gas coming out of solution in your bloodstream, okay? So it's a really specialized circumstance, okay, but it is something that can affect us. So in the water, to get to that level of pressure where that sort of thing can happen, you don't have to go that deep. You only have to go about 18 feet underwater. It's not very deep at all to have that sort of a risk sort of occurring, right? In the air, you would have to go a lot higher to have a change in pressure. So when has anybody, okay, more common thing, this is probably going to sound like a different question, but has anybody ever flown? Right? We all probably have been on an airplane at some point in our life. So if you fly, what happens is that the cruising altitude of an airplane, a big jetliner, is close to 30,000 feet. Obviously that varies a little bit depending on a number of things. But it's close to 30,000 feet elevation, right? So that's really low pressure. So what they have to do when you're on an airplane is they have to pressurize the cabin, okay? So now you're kind of in the same situation that you're in when you're scuba diving. You're breathing pressurized gas, okay? I know at the beginning of every airplane flight, they give a safety briefing that you probably all ignore. I know I do, all right? But when they give that safety briefing, they talk about the oxygen mask that will drop from above. But then that's in the event of what? Say it? In the event of cabin depressurization, right? They don't say in the event of oxygen loss. They say in the event of a loss of cabin pressure, right? Does that sound familiar now, right? I know you've ignored that your whole life. But in the event of a loss of cabin pressure, because what happens, right? You're breathing pressurized gas because you're in a big tube that's been pressurized at 30,000 feet. And now all of a sudden, boop, there's a loss of cabin pressure. Again, it's like opening a can of 7-Up in your bloodstream. All that pressurized gas stands the risk of coming out of solution. and you can get the bends actually from that, okay? So what you do is they want you to start breathing pressurized gas again as quickly as possible to help you avoid getting the bends, right? So, pretty cool, huh? Bet you didn't know that, right? It's not the oxygen, it's the bends that actually can put you at risk when an airplane loses cabin pressure. So now when you see your flight attendant or your pilot on your next flight, you can say, I don't want to get the bends, and they're going to be really impressed that you know that, because that's what it's from. Not that any of us, well, some of you might, but it's highly unlikely any of us will ever experience this. Space flight is the same thing. When astronauts do spacewalks, they stand the risk of getting the bends because they're going from a pressurized space station or space shuttle into space where there's no pressure at all. Again, they stand the risk of getting the bends as well. So there are a couple of very, very specialized instances in our lives where solubilized gas under pressure might become an issue for us, but generally speaking, it's not too big of a deal, right? Solubility of the gas, okay? I'm gonna cut to the chase on this one. I'm not gonna go into it a whole lot, but I'm just gonna tell you oxygen is not very soluble at all, okay? Oxygen has a really low solubility in solution. So oxygen's solubility in solution is pretty low. So at body temperature, at 37 degrees Celsius, which is body temperature, oxygen solubility is about 4 to 5 milliliters per liter of blood. Okay. Only 4 to 5 milliliters of oxygen can dissolve in 1 liter of blood. So that's really low. To give you an idea of how low that is, how many liters of blood do you have? About 5, right? About 5 liters of blood, right? So let's just go ahead and go on the high end of this and say that you're dissolving 5 milliliters of oxygen in 1 liter of blood. That gives you a total at any given time of 25 milliliters of oxygen in your entire bloodstream. Okay, that gives you 25 milliliters of oxygen in your entire bloodstream. If we assumed that all of that oxygen could be transferred to your tissues, which it can't, and I'll explain why a little bit later on this morning, but if we assumed that all of it could be, you would need to pump blood at a way higher rate than you're able to do. Because when you're exercising, your body needs about 4 liters of oxygen per minute. Okay? When you're exercising, your body needs about 4 liters of oxygen every single minute. So if we were to kind of take that out, right? 25 milliliters of oxygen in your bloodstream. So if you cycled that out, your cardiac output would have to be really, really high to be able to get enough oxygen to your tissues to survive. I think that comes out to around 2,500 pounds of blood that you have to circulate every single minute in order to make it. So the bottom line is you can't dissolve enough oxygen in your blood in order to meet your metabolic needs. Your cells need a lot of oxygen, and we can't dissolve enough oxygen in our blood in order to do that. So we had to evolve something else. What did we evolve to help us carry more oxygen in our blood? Hemoglobin. Yes, we evolved hemoglobin, right? So solubilizing the gas doesn't work. We do carry a little bit of oxygen solubilized in our blood, but for the most part, that oxygen is all bound to hemoglobin. Okay, so let's talk a little bit about hemoglobin. Hemoglobin is found in your red blood cells. Okay, so our options here are dissolved blood plasma. We said that doesn't work, right? So our next option is going to be to evolve hemoglobin. And that does work, okay? In fact, it works really, really well. Human evolution has had some fails, but hemoglobin is not one of them. It's a pretty cool protein. So we're going to talk a little bit about hemoglobin. Okay, so let's go ahead and spend some time talking about this amazing protein that will absolutely change your life. So hemoglobin is found inside of our red blood cells. And it has this really unique structure. We talked about the structure a little bit in our last unit, but it's made up of essentially 4 heme groups or globin groups, and each one of them has an iron sort of at the center of it. So it kind of looks like this structure like this. Right? We kind of have something that looks like this. So we've got this sort of thing called a heme group at the center of each one of these. We're not going to go into the molecular structure of it a whole lot, except to say that it's there. And it has iron at the structure. And then surrounding that heme group is this 3-dimensional protein that sort of holds it in place, right? So there's this chain of amino acids that's going to hold each one of these in place. Right, this globular protein. There are 4 total globular proteins, and you know, as we said a little bit ago, these represents a quaternary structure for a protein, which is fairly rare because we don't typically see proteins this big. But remember, you have a lot of these, enough to where you can actually fit approximately 1 billion oxygen molecules per cell. I've drawn these in different colors for a couple of reasons. The first is mostly clarity, just so they don't blend together too much. And the second is that they are actually 2 subunits with different names. They have the exact same function, but they do come from different genes in our DNA. So 2 of these are going to be what we call alpha subunits. So adult hemoglobin is made of 2 alpha subunits, and it's made of 2 beta subunits. Okay, so those are the subunits that are going to make up adult hemoglobin. And if you are picking up on the nuance here, you can bet that we're going to talk about non-adult hemoglobin in a little while, right? So adult hemoglobin is made up of 2 alpha and 2 beta subunits. So as we said when we talked about the blood, and we talked a little bit about red blood cells, hemoglobin does bind oxygen. Each one of those 4 heme groups is able to bind oxygen, a single oxygen molecule. So this particular hemoglobin molecule that we have up here, this can bind 4 total oxygen molecules, right? So there's a room right here for an oxygen, there's room right here for an oxygen, here and here. It is that iron that gives our blood its characteristic red color. Again, all you have to do is you have to go to Utah or Sedona, Arizona, or something like that, and see the red rocks. There's a high iron content in that rock, and it gives that rock a red color, right? That's that same characteristic that gives our blood its red color. Heme groups do vary a little bit across the animal kingdom, okay? So, but they always have some sort of a heavy metal as part of that heme group, okay? Does anybody know what horseshoe crabs are? You know what horseshoe crabs are? Horseshoe crabs are great, right? Horseshoe crabs are these little, they have them on the East Coast. We don't have them on the West Coast, but they are these very, very primitive things. They look like these little armored horseshoes that come up out of the ocean. They're pretty cool little animals. So they have a unique hemoglobin. Instead of having iron at the center of each of their heme groups, they actually have a molecule of copper. okay? So copper is the primary metal at the center of their heme groups. Their hemoglobin works in the same way that ours does, but they have copper at the center of their hemoglobin. Does anybody know what color copper turns when it's oxidized?
Green.
Kind of a greenish blue, yeah, exactly, right? If you ever worn a piece of copper jewelry or something like that, you'll know that at the end of the day, your skin is this greenish blue color, right? So yeah, copper has this kind of greenish blue color to it, mostly green, but with a little bit of blue. That's what color their blood is, okay? They have this kind of greenish blue blood. It almost looks like something out of a sci-fi movie. It's really, really strange. Other organisms have other colors of blood depending on the heavy metal that we find at the center of their heme group. Okay. The ones with Metallica at the center of their heme group, you don't even want to know what color their blood. I'm just kidding. I can't think of another heavy metal band off the top of my head. I know there's a million of them out there. And if you're a fan, you would probably argue that that's not truly heavy metal, but that's all right. Where's Aria when I need him? He would skewer me over that statement. All right. So anyway, if you know, you know. All right. So anyway, we happen to have iron. Okay. And the best way to sort of envision this structure, right, is these protein chains are these big globular three-dimensional things, right? In that heme group, it's kind of like suspended in the middle of it, right, with that iron right at the middle, right? But imagine that you've got this big three-dimensional spider web, and you've got this heme group right in the middle of it, just kind of suspended in it, right? Like it's literally being suspended in place, like hanging in the middle of this, right? So what happens is when oxygen kind of comes in, pretend this is oxygen, it kind of comes in and binds to that heme group shifts just a little bit, okay? Now it is a reversible binding, okay? So oxygen unbinds just as easily as it binds, and then the heme group goes back to its original position. Oxygen binds, shifts just a little bit, oxygen unbinds, it goes back just a little bit. Just that little shift that we see in that heme group, okay? What that does is it causes that heme group to reflect light just a little bit differently, whether oxygen is bound to it or whether oxygen is not bound to it, okay? And that's what accounts for that difference in the shade of blood that we see when we look at oxygenated blood versus deoxygenated blood. Just that little tiny shift causes a slight difference in the reflection of light, and it's a different shade of red when you see it, okay? So it's kind of an interesting thing. But as I said, hemoglobin does bind reversibly to oxygen. So we typically abbreviate hemoglobin as HB. It's way faster than writing it out, on and on and on. So we typically abbreviate it as HB. It binds with oxygen reversibly. So the reaction looks something like this. HbO2 is what we would call it when it's bound to oxygen. We call this oxyhemoglobin. Okay, call it oxyhemoglobin. Okay. All right. So as we said this morning, it's really earlier this morning and on Wednesday, it's really the concentration gradient that determines whether or not oxygen moves on to hemoglobin or off of hemoglobin. But let's go ahead and look at the relationship between the amount of oxygen available and how much of it actually binds to hemoglobin. So this is, in my opinion, this is one of the cooler things in all of biology. I think this is just a neat relationship. So I want to spend a little bit of time on this today. I need more boards. I need one of those ones that goes up, and then I have another one behind it, so I can keep it all. All right. So what I want to do is I want to spend a little bit of time with some graphs today. We've been graphed efficient lately in this class. but it's time to get caught up. So we're gonna spend some time with some graphs today. So what I'm gonna do is I'm gonna label my axes. We're gonna draw this graph several times today. So on the y-axis is the percent saturation of hemoglobin. Okay, so the percent saturation of hemoglobin. So what we got now is we've got a sample of whole blood, right? We've got a sample of blood. And in that sample of blood, you have way too many hemoglobin binding sites to count, right? Lots and lots and lots and lots, okay? So 0% saturation would mean that there is no oxygen bound to any of those binding sites, right? Okay? 100% saturation would mean that there's oxygen on every single one of those binding sites on every hemoglobin molecule. It's completely saturated. That sample of blood cannot hold any more oxygen. Does that make sense? Okay. So we go from 0 up to 100. Okay. And then on the x-axis, we're going to get oxygen partial pressure. Okay. So PO2. Remember, we said that if you are having a hard time with this, kind of visualizing partial pressures of gases, that's okay. Just think about this as an oxygen availability, right? So a high PO2 means that there's not that there's a lot of oxygen present. A low PO2 means that there's not a lot of oxygen present. Okay. Okay. So we do have some numbers though that we can do. We had on the board at the end of class on Wednesday. We put some numbers on these. Does anybody remember what we said alveolar PO2 is? That drawing that we had on the end of class on Wednesday. What was it? 100, yeah, right? It was 100 millimeters of mercury, right? So let's go ahead and put that on our x-axis. We're going to go ahead and put 100 millimeters of mercury. And this is alveolar PO2. And then, what did we say it was in a cell from that drawing? About 40, right? Yeah, it was about 40 millimeters of mercury. So I'm going to come over here and I'm going to put 40 down here as well. This is going to be in a cell, in a resting cell. Cool. Okay, so we've got a couple of numbers on there. On our x-axis, we have two physiologically relevant numbers. We have the oxygen availability that we would encounter in a cell, somewhere in our tissues. And then this is about as high as we're going to get. This is our alveolar PO2. So this is what you would measure when you take that breath of air, right? That's your PO2 in your alveolus. Okay. So let's go ahead. I'm gonna put some numbers on here, and we'll talk about them. So give me just a second to draw this curve, and we'll kind of go from there. I am going to put a 50 on here, and I'm going to put a 75 on here, and I'll explain why I'm putting those numbers. Besides the fact that they're nice round numbers, I'm putting them there for a reason. So I'll explain this here in just a minute. Okay, so our curve that we have looks something like this, okay? This curve is called a hemoglobin oxygen dissociation curve, okay? It's called a hemoglobin oxygen dissociation curve. That's what it's called. So let's go over some of the key aspects of this, okay? Let's go over some of the key aspects of this curve, and then we'll talk about it. So, all right, so let's go ahead and talk first about what's happening in the alveoli, all right? So over here, I'm over here at alveolar PO2, okay? If I were to start here, and I wanted to go see what my percent saturation is, I can draw a line going straight up, all the way until I intersect the dissociation curve. And then I can take that all the way over should be mostly straight. I'm sorry, I'm not the straightest person at 8 o'clock in the morning, the straightest line drawer at 8 o'clock in the morning. But we did pretty well for ourselves. I can draw it over, okay? Am I at 100%? I'm not, right? How many of you guys have ever used a pulse oximeter? We have one in lab. We're going to use it today a little bit if we have to remember. You used a pulse oximeter. So you've got a healthy, well-ventilated adult. What type of number would you expect to see on that? Higher 90s, right? 96, 97, 98%, right? 100%? Maybe for a split second, but not normally, right? Yeah. It's not something that normally happens, right? So for a normal, well-ventilated adult, we're looking at somewhere around 90, about 98-ish percent, right? Say 98% or so percent saturated, okay? Why are we not getting to 100%? Okay. Well, it turns out that hemoglobin's kind of a funny molecule. It's really good at binding to oxygen, but it behaves a little bit funny. You'll notice also that this curve is definitely not linear. It definitely has an S shape to it, right? It's what we call a sigmoid shaped curve. Right? So let's spend a couple of minutes kind of interpreting that curve and look at what it means. So hemoglobin has, I talked about the fact that heme group kind of changes conformation a little bit. It also has these molecular bonds that sort of hold those four subunits together. And when there's no oxygen bound to hemoglobin, it's held in what we call a tense state. Okay, tense meaning like really bound up and really, really tight. Okay, so the best way to sort of visualize that is to think about like pretend you've got like a big bath sponge, right? Like one of the big ones, and you scrunch it up into a ball as tight as you can get it, and you wrap a rubber band around it a whole bunch of times to hold it into place. Right? You envisioning that? Right? So that's what hemoglobin is kind of like when there's no oxygen bound to it. Okay? And imagine that you've wrapped that rubber band around that sponge enough times that you could take it, a scissor, and you could cut that rubber band in one place. Okay? What that sponge might do is it might a little bit, but the rubber band is still wrapped around enough times that it still holds it mostly in place. Okay? And then you cut the rubber band again, and it pops out a little bit more. Okay? And then finally, that third time you cut that rubber band, and the whole thing just, and the sponge pops out, right? Okay? It's kind of what hemoglobin does. Okay? The first oxygen that we put on hemoglobin only causes it to relax just a tiny little bit. Okay? And then the second one binds and it relaxes a little bit more, and then the third one binds and the molecule becomes more relaxed, and it's easier to add oxygen onto hemoglobin. Okay? So we see that with the curve. Okay? We see that with the curve. Remember that my x-axis here is oxygen, right? PO2. So as this goes along, there's more and more oxygen available, and this is percent saturation. So if we only wanted to look, let's just pretend that we're only looking at this part of our graph. Pretend the rest of it doesn't exist, okay? And I look at the slope of that graph. I know we're going down the math road here, and I'm sorry about that. But we're looking at the slope of that graph, right? I see that it's not a very steep slope. So what that tells me is I can add a lot of oxygen or have big changes in oxygen down here, and I don't see a very big change in percent saturation of oxygen, okay? But once I've added a couple, look at what happens to the slope of the graph. It gets way steeper. That means very small changes in oxygen result in way more oxygen being bound to hemoglobin, okay? And then up here at the very top of the graph, it levels out again. Those last couple of oxygen are really, really hard to add to hemoglobin. Okay, they're not very easy to add to hemoglobin, which is why with PO2s like alveolar PO2, we have a really hard time getting to 100% saturation. Okay, so it's a cool molecule, but it behaves a little bit on the funny side. Okay. Okay. The other thing that I want to kind of point out here is I want to point out what the percent saturation is in a resting cell, right? Because this is where we said earlier today that what we're doing here is we're transferring oxygen from the blood to the tissues, right? And when we studied the cardiovascular system, we had a name for that blood that was coming back from the tissues to the heart, right? What was the name that we gave that blood that was in the inferior and the superior vena cava? Deoxygenated, right? Remember I said I don't like that term? Okay. Here's why. All right. Let's look at what happens in our resting cells. Let's draw our dotted line all the way up to our graph. And let's go over. Right? If we look at at a PO2 of 40 millimeters of mercury, which is what we encounter in our tissues, and I take that up to my line and I come over here, I see that blood, 75% of those binding sites still are occupied by oxygen. So that blood is not deoxygenated, right? Deoxygenated implies no oxygen, right? But 75% of those binding sites are still occupied by oxygen. Why would that be? Why would we just not dump all of the oxygen in our tissues and then go back and get more? Anybody have any ideas? Send it back through your lungs, get more oxygen, and send it back out. If I got rid, if I got rid of all of the oxygen in my blood, okay? If I got rid of all of the oxygen in my blood, just dumped it all in my tissues and dropped this down to 0% saturation, if there was a situation like I was just in where I can't reoxygenate my blood, there's nothing else to send to my tissues. But because I maintain a pretty good percentage of oxygen bound to hemoglobin, if I have to send that blood through my circulatory system again, I have oxygen that I can still drop off to my tissues, okay? So it obviously isn't gonna last forever, but it's better than nothing, right? So deoxygenated blood, as we know it, as we study it, is not really deoxygenated at all. It actually has quite a bit of oxygen in it. It just has less than the blood leaving the lungs, okay? So, does that make sense? It's kind of cool, right? All right, I'm gonna have to hold my breath later on today to show you some more stuff, so I need to get practice for that. All right, so anyway, it's kind of an interesting curve. It's an interesting relationship. For what it's worth, if this curve was linear, this number would be a lot lower. So it's an interesting one. So I talked about how it's kind of hard to start putting oxygen onto hemoglobin, and then it's kind of hard to put those last few on. The same thing is true when we start to take oxygen off of hemoglobin, the curve is reversed, and it's kind of hard to take those first few off, and that actually ends up benefiting us, right? So it's kind of an interesting one. Okay, so the next thing I want to talk about is the shifts that we experience in this curve. And the shifts that we experience in this curve have to do with the affinity of oxygen for hemoglobin, okay? And I talked about how we experience, it's kind of hard to put the first few on. It's kind of easy to put the middle ones on, and it's harder to put the last few on, right? But let's take a step back from that and let's think more globally about the affinity of hemoglobin for oxygen. When I talk about that, what am I talking about? Like if I use that term affinity, what comes to mind? If you have affinity for something, means that you like it, right? Yeah, right? So when I talk about the affinity of two molecules for each other, in this case, hemoglobin and oxygen, I'm talking about how tightly they bind to each other, okay? If they have a high affinity for each other, then it binds really tightly and really easily, okay? That's kind of a key. If the affinity isn't so high, they don't bind as tightly, they don't bind as easily to each other, okay? So let me ask you, all right, which do you think would be better? Would you rather have a hemoglobin with high affinity for oxygen, or would you rather have a hemoglobin with a lower affinity for oxygen? Okay. How many guys say high? Okay. How many guys say low? How many guys don't know? Okay, fair. That's all right. Okay. So we're looking at two places, essentially, right? We're looking at what happens when we load oxygen onto the hemoglobin at the lungs, right? And we're looking at what happens when we unload oxygen at the tissues, right? So, for those of you who said high, all right, can you explain why? Does somebody want to try to maybe explain why you said you'd rather have a hemoglobin with a high affinity for oxygen? Even if you're wrong, we're all friends. Yeah. Okay. For sure, right? Because oxygen's good. We need it, right? Yeah, absolutely. Okay. What about low? A couple hands went up when I said you'd like to have low. Does anybody want to try to explain that one?
That way, the hemoglobin wouldn't give up the oxygen.
So low means that it actually gives it up easier. It's harder to bind, it gives it up easier. Okay. Okay. No, you got no explainers? Okay, that's all right. Okay. So let's think about this a little bit, right? Okay, let me give you a little bit more and let's see if any minds change. Okay. So at the lungs, right? Cody gave a good explanation, right? He said that we need oxygen, and you want to try to get as much as you can, right? So if you're in your alveoli, it'd be really great maybe to have hemoglobin with a high affinity for oxygen, right? Because you can get as much as you can get from the air, right? Cool, right? Okay. What about at the tissues, right? Remember, a high affinity oxygen or a high affinity hemoglobin means that it binds really, really tightly. So it binds tightly, but that also means that it doesn't unbind easily, right? So at the tissues, you'd actually want it to be lower, right? Because you want to give as much oxygen to your tissues as possible, right? Does that make sense? So we have two different places, right? We have the lungs, the alveoli, and we have the tissues. And does anybody want to, so at the lungs, which one would you want? High affinity, low affinity? How many of you want high affinity at the lungs? I do too. Yeah, I do too. How many of you want low affinity at the tissues? Me too. I want that too. Guess what? You can't have both. Okay? You get one. It's the way that it works. Your hemoglobin is your hemoglobin is your hemoglobin. We can't change its affinity in flight. It doesn't work that way, right? So it's too bad, so sad. You got to pick one. So probably somewhere in the middle is probably our best bet, right? The thing is, there's not really an easy answer to that question, right? So I don't really blame you guys for thinking about it. There's not one single best answer. Nobody was wrong in terms of your opinion, even the first time around, nobody was But we have to understand that there is a trade-off that occurs. If I have super high affinity hemoglobin, it's going to be easy to get that oxygen bound to it at my lungs. It's going to be kind of hard to get it off at the level of the tissues, okay? If my hemoglobin affinity is a lot lower, it's going to be really easy to unload it at the tissues, but I'm not going to get as much on at my lungs, okay? Okay. So the good thing, I suppose if there is a best answer, I would want a low, okay? I would want low affinity hemoglobin. Let me explain why, okay? Let's say that I do have low affinity hemoglobin, all right? I want it to unload easily here. And let's say that I'm not getting as much on right here. In fact, let's say that I don't have as much oxygen here, right? Let's say that I have a patient with chronic obstructive pulmonary disorder, okay? And they're not ventilating their lungs as well as they should, or maybe they've got lung cancer and they've got a restrictive pulmonary disorder, right? And they're just not getting as much air into their lungs. Let's say that their alveolar O2, CO2, is 90 millimeters of mercury, okay? Let's see what happens then. Let's follow that up to our graph. Oh, look, still 98% saturated. Isn't that amazing how that works, right? So I can actually suffer drops in my alveolar PO2 and not really suffer a drop to my percent saturation of oxygen. And that has a lot to do with the nature of the curve and the fact that it levels out so much at the top, right? So if it was linear, it wouldn't work that way. So it's kind of a neat one. So I think I'd rather have a low affinity hemoglobin. I think I would. I've never taken a side in this argument before. You guys are the first class I've ever tried to take a side in. So, but I think I would. I think I'd like a lower affinity hemoglobin because I know I can still get quite a bit of oxygen on just because of the nature of the curve. So I'm leveling the knowledge that I've got here. All right. But there are a couple of things that do change the affinity of this curve. I'm going to run out of time here, but let's just go through this really fast, and then I'm going to have to find some time to finish lecturing on this stuff. Okay, so there are some factors that can actually take this curve and shift it either to the left or to the right. Okay, so there are some factors that can take this curve and shift it over to the left, or shift it to the right. It might have been lyrics to a Jimmy Buffett song, right? Swim to the left, swim to the right. No Jimmy Buffett fans in here. All right, let's go ahead and talk about this really quick. So, and then we'll end with this part, and then we'll have to continue this discussion at a later date. There's never enough time to talk about hemoglobin. All right, so when we shift the curve to the right, and the right is the direction I'm most concerned about. The left is okay, but the right is the one I'm more concerned about, or the more interested in, I guess I should say. When we shift the curve to the right, okay, what are we doing? Okay, well, what we're doing is we're changing the affinity of hemoglobin for oxygen, and we're actually reducing the affinity of hemoglobin for oxygen. Let me explain and prove to you why we're reducing that affinity. Let me get rid of some of my lines here. There's one more measurement that we need to take here. One more thing that we need. Because we got to have a way to talk about these, right? And put numbers to these curves so that they make sense. The number I'm interested in right now is called the P50. P50 is defined as the partial pressure of oxygen, the PO2, at which hemoglobin is 50% saturated with oxygen. CO2, at which hemoglobin is 50% saturated with oxygen. Okay, so if we my arrow right where I need to be. Let's go ahead here, that one, let's take a look. So here's my 50% saturation, okay? I'm gonna draw a line all the way over to my curves. And again, I'm only interested in the green one and the black one right now. I'm gonna ignore the blue one for a minute. So here I'm gonna draw a line down here, and here I'm gonna draw a line down here. Okay. So I see that this one is about 30 millimeters of mercury, and then this one is about 40 millimeters of mercury, right? So I have two curves. Each of them has a different P50. One of them is about 30 millimeters of mercury, the other is about 40 millimeters of mercury. So what does that mean? How do I interpret that? To get my hemoglobin to 50% saturation, I need more oxygen for the green curve, okay? That tells me that that curve is not binding as tightly to oxygen. I have to put more oxygen into the mix to get to the same level of saturation, okay? Does that make sense? To put more oxygen into the mix to get to the same level of saturation, okay? This one, I get to 50% saturation with just a small amount of oxygen. This one, I have to add more oxygen to get to the same level of saturation because it's just not binding as tightly. Okay. Now with that blue curve that I shifted to the left, I don't need very much oxygen at all, and I'm already at 50% saturation, meaning that the affinity is very, very strong. Okay. So a right shift means a reduction in affinity. Okay. A right shift, if we go to the right, that means a reduction in affinity. And if we go to the left, it means an increase in affinity. Okay. I know we haven't spent much time with graphs this semester, and now we're hitting you hard with them today. So this might take a little bit of working out over the weekend, and I understand you might have to work through this verbally a few times to make sure that you understand it. So I do want to talk about some factors that affect the affinity of hemoglobin for oxygen, because there are a few really important ones. And there are some ways that we can change this, mostly related to exercise, and we'll kind of talk about that a little bit later on. We're almost there though, okay? We're almost there, and then we'll be kind of wrapping up with some of the lecture material on the respiratory system. But we're getting close. We're through the hard part, all right? So today in lab, what we do want to do is we want to give a quiz in lab on the respiratory system anatomy. And then what we'll do is I want to talk about some of the kind of more practical things related to the respiratory system, specifically related to the amount of air that you can move in and out of your lungs and your ability to do that and things like that. If we have time, we'll come back and we'll try to finish this up. Otherwise, I'm going to have to do it on Wednesday of next week. So I was hoping to get done today, but you know, time.